Hello YouTube, my name is Neil. We're studying for the MCATs today, biology. We are in chapter 3, Embryogenesis and Development. Is it a boy or a girl? It's one of the most common questions asked of pregnant women. The suspense around learning the baby's sex never fails to excite family and friends. For centuries, members of the elder generations have offered advice on how to predict the outcome or even plan it. Dangle a needle over your belly by a thread. Does it swing side to side or in circles? In the modern world, needle dangling doesn't carry the same credibility as technology or technological advances like ultrasonography. This radiographic technique is performed by placing a probe that emits high-frequency sound waves near the tissue to be examined. The probe transduces an image onto a computer screen, which can be measured to determine gestational age, screen for multiple pregnancies or anomalies, and identify the baby's sex. So how early can we determine a baby's sex? All embryos begin female by default. That is, for a male fetus to develop, it must undergo not only masculinization via the gene product of SRY, but also defeminization. These processes occur or don't around 6 to 8 weeks post-fertilization. That said, prospective parents shouldn't expect any answers before 16 to 17 weeks without a blood test, because ultrasonography equipment does not have high enough resolution yet to give immediate answers. In this chapter, we'll continue the discussion from the previous chapter by beginning with fertilization, the formation of a diploid zygote, diploid zygote from the union of a sperm and an ovum. Will then follow development from this point until the birth of an autonomously breathing baby. We'll examine how the cells of a developing human divide and differentiate. We'll also explore some specific system differences that exist between developing fetuses and adults as we present an overview of the stages of pregnancy and birth. Let's write on here. Embryogenesis and development. Early developmental stages. In this first section, we explore development from the formation of a diploid zygote until neurolation, or the formation of the neural tube that will differentiate into the nervous system. Fertilization. As discussed in Chapter 2, a secondary oocyte is ovulated from the follicle on approximately day 14 of the menstrual cycle. The secondary oocyte travels into the fallopian tube where it can be fertilized up to 24 hours after ovulation. 24 hours after ovulation. Fertilization, shown in figure 3.1, occurs in the widest part of the fallopian tube called the ampulla. When the sperm meets the secondary oocyte in the fallopian tube, it binds to the oocyte and releases acrosomal enzymes that enable the head of the sperm to penetrate through the corona radiata and zona pellucida. Once the sperm comes into direct contact with the secondary oocyte's cell membrane, it forms a tube-like structure known as the acrosomal apparatus, which extends to and penetrates the cell membrane. Its pronucleus may then freely enter the oocyte once meiosis II has come to completion. So we're at meiosis II here. After penetration of the sperm through the cell membrane, the cortical reaction, a release of calcium ions occurs. Let's write that down too. Cortical reaction. Let's put calcium ions in there. These calcium ions depolarize the membrane of the ovum, which serves two purposes. Depolarization prevents fertilization of the ovum by multiple sperm cells, and the increased calcium concentration increases the metabolic rate of the newly formed diploid zygote. The now depolarized and impenetrable membrane is called the fertilization membrane. Twins. Can occur by two different mechanisms. Whoops. Dizygotic or fraternal. I bet dizygotic means identical. Form from fertilization of two different eggs released during one ovulatory cycle. Oh, this is fraternal. Dizygotic is fraternal. Dizygotic occurs from the fertilization of two different eggs released during one ovulation cycle by two different sperm. Each zygote will implant in the uterine wall, and each develops its own placenta, chorion, and amnion. These structures are discussed later in the chapter. If the zygotes implant close together, the placentas may overgrow each other. Fraternal twins are no more genetically similar than any other pair of siblings. Monozygotic or identical twins form when a single zygote splits into two. Because the genetic material is identical, so too will the genomes of the offspring. If division is incomplete, conjoined twins may result, in which the two offspring are physically attached at some point. Monozygotic twins can be classified by the number of structures they share. Monochorionic, monoamniotic twins share the same amnion and chorion. Monochorionic diamniotic twins each have their own amnion but share the same chorion. Dicryonic and diamniotic twins each have their own amnion and chorion. Each type of twin occurs. Which type of twinning occurs is a result of when the separation occurred. As more gestational structures are shared, there are more risks as the fetuses grow and develop. All right. So in the monozygotic class, you have conjoined twins. Then you have monochorionic. Monochorionic and you can have dichorionic and diamniotic. All right. Cleavage. After fertilization in the fallopian tubes, the zygote must travel to the uterus for implantation. If it arrives too late, there will no longer be an endometrium capable of supporting the embryo. In the process of moving to the uterus for implantation, let's just write cleavage down here. The zygote undergoes rapid mitotic cell divisions in a process called cleavage. Cleavage is rapid mitotic divisions. The first cleavage officially creates an embryo as it nullifies one of the zygote's defining characteristics, unicellularity. Okay, so zygote to embryo, 1 cell to 2 cells, I guess. Although several rounds of mitosis occur, the total size of the embryo remains unchanged during the first few divisions. As shown in figure 3.2, by dividing its progressively smaller cells, the cells increase two ratios, the nuclear to cytoplasmic ratio, the surface to area, and the surface to volume ratio. Thus, the cells achieve increased area for gas and nutrient exchange relative to overall volume. There are two types of cleavage, indeterminate and determinate. Indeterminate cleavage results in cells that can still develop into complete organisms. In fact, monozygotic twins have identical genomes because they both originate from indeterminately cleaved cells of the same embryo. Determinate cleavage results in cells with fates that are, as the term implies, already determined. In other words, these cells are committed to differentiating into a different type of cell. All right. So let's just write down that MC ratio, nuclear. And also surface area to volume. Let's also just put surface area to volume. Let's write them in. And then you have indeterminate versus all right. Blastulation. Several divisions later, the embryo becomes a solid mass of cells known as a morula. As shown in figure 3.3, the term comes from the Latin word for mulberry, which might help us grasp what an embryo at this stage looks like. Once the morula is formed, it undergoes blastulation, which forms the blastula, a hollow ball of cells with a fluid-filled inner cavity known as a blastocell. Should we draw a little drawing here? The mammalian blastula is known as a blastocyst. So this is the inner cavity. This is also called blastocyst. The blastula in mammals is a blastocyst and consists of two noteworthy cell groups, as shown in figure 3.4, the tropoblast and inner cell mass. The tropoblast cells surround the blastocell and give rise to the chorion and later the placenta, whereas the inner cell mass protrudes into the blastocell and gives rise to the organism itself. All right, we got the tropoblast layer. And we have the inner cell mass on the inside. Implantation. The blastula moves through the fallopian tube to the uterus, where it burrows into the endometrium. The trophoblast cells are specialized to create an interface between the maternal blood supply and the developing embryo. These tropoblastic cells give rise to the chorion, an extraembryonic membrane that develops into the placenta. The tropoblasts form chorionic villi, which are microscopic finger-like projections that penetrate the endometrium. As these chorionic villi develop into the placenta, they support maternal-fetal gas exchange. The embryo is connected to the placenta by the umbilical cord, which consists of 2 arteries and 1 vein encased in a gelatinous substance. The vein carries freshly oxygenated blood rich with nutrients from the placenta to the embryo. The umbilical arteries carry deoxygenated blood and waste to the placenta for exchange. So let's write some of this down. Trophoblast becomes the chorionic villi, where villi are finger-like projections. The umbilical cord consists of 2 arteries and 1 vein encased in gelatinous substance. All right. Until the placenta is functional, the embryo is supported by the yolk sac. The yolk sac is also the site of early blood cell development. There are 2 other extraembryonic membranes that require discussion: the Elantois and the amnion. The Elantois is involved in early fluid exchange between the embryo and the yolk sac. Let's write that down. Elantois. Ultimately, the umbilical cord is formed from remnants of the yolk sac and the Elantois. So these 2 things become the umbilical cord. The Elantois is surrounded by the amnion, which is a thin, tough membrane filled with the amniotic fluid. This fluid serves as a shock absorber during pregnancy, lessening the impact of maternal motion on the developing embryo. In addition to forming the placenta, the chorion also forms an outer membrane around the amnion, adding an extra level of protection. The anatomy of these structures is shown in figure 3.5. So the amnion is inside the chorion, which forms a placenta. We should also write that this forms the placenta. right? So the amnion is an internal layer. The chorion's the external layer. The placenta looks like it's here in this chorion layer, right? The amnion is this internal layer. And what did we learn becomes the umbilical cord? The Elantois and yolk sac become the umbilical cord. All right. Gastrulation. Once the cell mass implants, it can begin further developmental processes such as gastrulation, generation of 3 distinct cell layers. All right. 3 cell layers. The early developmental process up to that point are shown in figure 3.6. Much of our knowledge of development comes from the study of other organisms that have varying degrees of similarity to human development. In sea urchins, gastrulation begins with a small invagination in the blastula. Cells continue moving toward the invagination, resulting in elimination of the blastocell. To visualize this, imagine inflating a balloon and then poking one of the sides with your finger. If you keep pushing, eventually the rubber from one side of the balloon would come into contact with the other side. If the two membranes could merge, as occurs in development, this would create a tube through the middle of the balloon. In living things, the result of this process is called a gastrula. The membrane invagination into the blastocell is called the archenteron, which later develops into the gut. The opening of the archenteron is called the blastopore. In deuterosomes, such as humans, the blastopore develops into the anus. In protozoans, it develops into the mouth. That was a lot of information. Let's draw a little bit. Let's say that we have a blastula, and then eventually becomes pinched off into a blastopore. The blastopore is the opening. Blastocell is the inside. Blastula to gastrula. The membrane invagination into the blastocell is called the archenteron. So let's call this archenteron. And let's call this the blastopore. So this is going to become the mouth in humans. Or no, it's going to be the anus in humans. Anus. In protozomes, it develops into the mouth. I don't know what deuterostomes and protostomes are, but yeah, primary germ layers. Eventually, some cells will also migrate into what remains of the blastocell. This establishes 3 layers of cells called primary germ layers. The outermost layer is called the ectoderm and gives rise to the integument, including the epidermis, hair, nails, and epithelia of the nose, mouth, and lower anal canal. The lens of the eye, nervous system, including adrenal medulla, and inner ear are also derived from the ectoderm. Ectoderm, let's put epidermis in there. Middle layer is called the mesoderm and develops into several different systems, including the musculoskeletal, circulatory, and most of the excretory. Mesoderm also gives rise to the gonads, as well as the muscular and connective tissue layers of the digestive and respiratory systems and the adrenal cortex. So everything in the middle, all the organs, except the innermost layer is called the endoderm and forms the epithelial linings of the digestive and respiratory tracts, including the lungs, the pancreas, thyroid, bladder, and distal urinary tracts, as well as parts of the liver, are derived from the endoderm. So the guts, digestive and respiratory. Let me change what I said about the mesoderm. Specifically, it's musculoskeletal. Circulatory, excretory, most of the excretory systems. Oh yeah, epidermis and, more importantly, the nervous system. All right, ectoderm is the nervous system. Mesoderm is muscles and bones and circulatory systems, and the endoderm is digestive and respiratory. Differentiation. So how is it that cells with the same genes are able to develop into such distinctly different cell types with highly specialized functions? Primarily, it is by selective transcription of the genome. In other words, only the genes needed for that particular cell type are transcribed. Thus, in pancreatic islet cells, the genes to produce specific hormones, insulin, glucagon, or somatostatin, are turned on, while these same genes are turned off in other cell types. Selective transcription is often related to the concept of induction, which is the ability of one group of cells to influence the fate of their other nearby cells. This process is mediated by chemical substances called inducers, which diffuse from the organizing cells to the responsive cells. These chemicals are responsible for processes such as the guidance of neuronal axons. This process also ensures proximity of different cell types that work together within an organ. All right. Selective transcription. What else did we learn? Induction, which is dependent on inducers. Are inducers like hormones then? Or are they just simpler than that? Neurolation. Once 3 germ layers are formed, neurolation, or development of the nervous system, can begin. Remember that the nervous system is derived from the ectoderm. How then do cells originating on the surface of the embryo, ectoderm, end up inside the final organism? First, a rod of mesodermal cells known as the notochord form along the long axis of the organism like a primitive spine. In fact, small remnants of notochord persist in the invertebral discs between vertebrae. The notochord induces a group of underlying ectodermal cells to slide inward to form neural folds, which surround a neural groove, as shown in figure 3.7. The neural folds grow towards one another until they fuse into a neural tube, which gives rise to the central nervous system. At the tip of each neural fold are neural crest cells. These cells migrate outward to form the peripheral nervous system, including the sensory ganglia, autonomic ganglia, adrenal medulla, and Schwann cells, as well as specific cell types in other tissues, such as calcitonin-producing cells of the thyroid, melanocytes in the skin, and others. Finally, ectodermal cells will migrate over the neural tube and crest to cover the rudimentary nervous system. All right, that's also a lot of information. Let's draw. Let's draw a picture of this. All right. We got a neural plate. Got a neural groove. And neural folds. And then finally we have a neural tube. And a notochord. Did we cover all the words in there? Notochord forms along the long axis. Primitive spine. And the neural fold and groove form, which gives rise to the central nervous system. So this is going to be the neural tube is the central nervous system. The tip of the neural folds are the neural crest. These cells migrate to make the peripheral nervous system. Finally, ectodermal cells will migrate over the neural tube and crest to cover the rudimentary nervous system. Surface ectodermal cells. All right, I think we got it all in there. Problems in early development. Problems. Early development is a highly sensitive time during the development of a human being. During this stage, as the germ layers are forming, and then organogenesis, the production of organs. Let's write that down. Organogenesis begins, and then organogenesis, the production of organ begins. Teratogens may have far-reaching and highly detrimental effects. Teratogens are substances that interfere with development, causing defects or even deaths of the developing embryo. Teratogens. However, each teratogen will not have the same effect on every embryo or fetus. It is believed that the genetics of the individual embryo influences the effects of the teratogen. In addition to genetics, the route of exposure, length of exposure, rate of placental transmission of the teratogen, and the exact identity of the teratogen will also affect the overall outcome. Some common teratogens include alcohol, prescribed drugs, viruses, bacteria, and environmental chemicals, including polycyclic aromatic hydrocarbons. Polycyclic aromatic hydrocarbons? Is that like gas? Teratogens. Any kinds of poisons, including viruses and bacteria. So it's basically any foreign substance almost. As well as pathogens. Poisons as well as pathogens. Alright, in addition to teratogens, development can also be influenced by maternal health. Certain conditions may cause changes in the overall physiology of the mother, resulting in overexposure or underexposure of the embryo or fetus to certain chemicals. For example, diabetic mothers with hyperglycemia, high blood glucose, can have poor birth outcomes. Overexposure to sugar in utero can lead to a fetus that is too large to be delivered and who suffers from hypoglycemia soon after birth due to synthesizing very high levels of insulin to compensate. Maternal folic acid deficiency may prevent complete closure of the neural tube, resulting in spina bifida or anencephaly, in which parts of the nervous system are exposed to the outside world or covered with a thin membrane. However, like teratogens, maternal health issues can have variable effects on the developing fetus. Spina bifida may be so severe as to result in profound disability, or may be completely asymptomatic and only detected by a tuft of hair overlying the area. Overall, trends and associations can certainly be found between various environmental conditions and genes during development. However, it is somewhat unpredictable and highly variable. All right. Let's see. Let's write down hyperglycemia in the parent can lead to hypoglycemia in the child. Is that what I understood? For example, diabetic mothers, hyperglycemia can have poor birth outcomes. Overexposure to sugar in utero can lead to the fetus that is too large that can be delivered and who suffers from hypoglycemia soon after birth due to synthesizing very high levels of insulin to compensate. In mother can lead to hypoglycemia in child. Let's write compensation. Insulin compensation. And folic acid. Low folic acid can lead to spina bifida as well as anencephaly. All right. What is the difference between determinate and interdeterminate cleavage? A determinate is any of the cleavages that result in, still the same cell types. Indeterminate is when they start differentiating, or determinate is when they start differentiating. Indeterminate is when any of the cells can still become any of the other cells. From zygote to gastrula, what are the various stages of development? Zygote turns into an embryo. During cleavage, and then you end up with a morula, which through blastulation becomes a blastula. Then comes implantation into the endometrium. By the end of implantation, you are left with a, I don't know, I guess it's still an embryo. But you have a sac, and you have a placenta, and a chorion and an amnion. Through gastrulation, are we ending the gastrula? Yeah. The blastula turns into a gastrula through gastrulation, which is when the invagination of the blastopore, or of the blastocell. And then you get 3 different cell layers. So it's up to gastrula. So I guess we'll end there. During which stage of development does implantation occur? Implantation occurs, I guess it's got to be during blastulation? Or is it during gastrulation? I think it's during blastulation. We'll find out in a minute. What are the primary germ layers and what organs are formed from each? You have the endoderm, which makes the epidermis and nervous system. The mesoderm, which makes the musculoskeletal system, the circulatory system, as well as the excretory system. The endoderm makes the digestive and respiratory systems. What is induction? I guess induction is... what was induction? The process by which similar cells influence each other. They release transducers that influence nearby cells to, become the same cell type as them, express the same proteins as them. What tissues do neural crest cells develop into? Neural crest cells develop into the peripheral nervous system, I believe. Let's look at the answers real quick, at least for 3. What stage does implantation occur? Implantation occurs during the blastocyst stage. Right. That makes sense. It was right before gastrulation. It's still in the blastulation phase here. All right. Mechanisms of development. As described earlier, cells undergo determinate cleavage to commit to a particular cell line, and inducers can be used to communicate from one group of differentiating cells to another. In this section, we take a more specific look at the biochemical properties of these developmental mechanisms, as well as a few selected others. Cell specialization. An adult human being is composed of approximately 37 trillion cells. Is that right? 37 trillion? Let's write that down. 37 trillion. These cells are organized into tissues, which form organs within organ systems. In order to create an organism as complex as a human being, each cell must perform a specialized function. In addition, the cells in an organ must be organized such that the organ can function properly. For example, the pancreas must create both digestive enzymes, trypsin, carboxypeptidases A and B, pancreatic lipase, and others. And endocrine hormones, insulin, glucagon, and somatostatin. The cells that synthesize digestive enzymes must be located such that the cell products can enter ducts to ultimately empty into the duodenum. Likewise, the cells that synthesize endocrine hormones must be located near a blood vessel to put their products into systemic circulation. In order to accomplish this, the cells must go through 3 stages: specification, determination, and differentiation. There's just some interesting facts there. That the pancreas makes. Trypsin, carboxypeptidase AB, and endocrine hormones. It also makes endocrine hormones like insulin, glucagon, somatostatin. I know these, we'll probably get into these more in maybe biochemistry or something, but these are just interesting, good to know. Specification/determination. The initial stage of cell specialization is specification. In which the cell is reversibly designated to a specific cell type. This is followed by determination. Determination, which was previously defined as the commitment of a cell to having a particular function in the future. Prior to determination, the cell can become any cell type, even if it has already gone through specification. What? The initial stage of cell specialization is specification, in which a cell is reversibly designated to a specific... Oh, okay, reversibly. So I'm going to remove that and put reversible designation. So specification is a reversible designation, whereas determination is as the commitment. All right, so we'll put down commitment. After determination, the cell is irreversibly committed to a specific lineage. There are multiple pathways by which determination may occur. If during cleavage, the existing mRNA and protein in the parent cell has been asymmetrically distributed between the daughter cells, the presence of specific mRNA and protein molecules may result in determination. Determination may also occur due to secretion of specific molecules from nearby cells. These molecules, also called morphogens, may cause nearby cells to follow a particular developmental pathway. Determination is a commitment to a particular cell type, but note that the cell has not yet actually produced the products it needs to carry out the functions of that cell type. This is the goal of differentiation. Let's put morphogens. Differentiation. After a cell's fate has been determined, the cell must begin to undertake changes that cause the cell to develop into the determined cell type. This includes changing the structure, function, and biochemistry of the cell to match the cell type through differentiation. Cells that have not yet differentiated, or which give rise to other cells that will differentiate, are known as stem cells. Stem cells exist in embryonic tissues as well as adult tissues. The tissues a particular stem cell can differentiate into are determined by its potency. Cells with the greatest potency are called totipotent and include embryonic stem cells. Totipotent cells can ultimately differentiate into any cell type, either in the fetus or in placental structures. After the 16-cell stage, the cells of the morula begin to differentiate into two groups, the inner cell mass and the trophoblast cells. So let's also write down that the 16-cell morula So since you have a 16-cell morula that is undifferentiated, my question is, could each of those 16 cells make like a twin? Could you have like potentially 16 twins anyways, that's something for you to think about. All right, so after the 16-cell stage, the cells of the morula begin to differentiate into two groups, the inner cell mass and the trophoblast cells. After a few more cycles of cell division, these totipotent cells start to differentiate into the three germ cell layers. At this stage, the cells are said to be pluripotent. All right, so let's put down that the three germ layers, I guess you could call them germ cells, germ cell layers. Are pluripotent. So we had the totipotent up here, pluripotent over here. These cells can differentiate into any cell type except for those found in the placental structures. All right. Finally, as the cells continue to become more specialized, they are said to be multipotent. Multipotent stem cells can differentiate into multiple types of cells within a particular group. For example, hematopoietic stem cells are cells that are capable of differentiating into all of the cells found in blood, including the various types of white blood cells, red blood cells, and platelets, but not into skin cells, neurons, or muscle cells. While we use all of these different terms to describe potency, it is important to recognize that potency is a spectrum, not a series of strict definitions. Also note that stem cells exist not only in embryos, but also in adults. We have stem cells that give rise to skin and blood and the epithelial lining of the digestive tract, among others. Multipotent. For example, can become just as an example, hematopoietic stem cells can become white blood cells, red blood cells, and platelets, but not other kinds. That's an example of multipotent cells. There are distinct classes, I guess. Over the last few decades, stem cell research has been a hotly contested issue. While harvesting of embryonic stem cells, highlighted in figure 3.8, ultimately results in destruction of the embryo, it is thought that these cells could be used to regenerate human tissues, including the spinal cord following injury and cardiac myocytes following a heart attack. There are also immunologic concerns, as transplantation of stem cells of a different genetic makeup would evoke an immune response, resulting in rejection. In addition, pluripotent cells may not always differentiate into the desired tissue once implanted and may even become cancerous. In order to address this controversy, many researchers have begun investigating adult stem cells. At best, these cells are multipotent, able to differentiate into only a few different cell types. Researchers may take adult stem cells and use various transcription factors to increase potency in these cells. One of the potential advantages of this is that a stem cell can be taken from a patient, usually from blood, bone marrow, or adipose tissue, induced to become a different tissue type, and then implanted into the same patient. This offers reduced risk of rejection of foreign tissue. However, it is challenging to induce differentiation into the correct cell type, and most organs have a complex structure that depends on a number of different cell types, each of which requires different signals. Research is ongoing and holds promise despite limited success so far. Induced pluripotency. Cell-cell communication. The determination and differentiation of a cell depends on the location of the cell itself, as well as the identity of the surrounding cells. The developing cell receives signals from organizing cells around it and may also secrete its own signals. As discussed previously, surrounding tissues induce a developing cell to become a particular cell type via inducers. The term inducer may also refer to the cell secreting the signal. The cell that is induced is called a responder, responsive cell, to be induced. A responder must be competent or able to respond to the induced signal. They have to be competent. Inducer, responder. I think we can remember that. We can keep those apart, right? Cell-cell communication can occur via autocrine, paracrine, juxtacrine, or endocrine signals. Oh, there we go. Autocrine. signals act on the same cell that secreted the signal in the first place. Paracrine signals act on cells in the local area. Juxtacrine signals do not usually involve diffusion, but rather feature a cell directly stimulating receptors of an adjacent cell. Finally, endocrine. Endocrine signals involve secreted hormones that travel through the bloodstream to a distant target tissue. Distant and targeted. Inducers are commonly growth factors. Which are peptides that promote differentiation and mitosis in certain tissues. Most growth factors only function on specific cell types or in certain areas, as determined by the competence of these cells. In this way, certain growth factors can code for particular tissues. For example, PAC6 is expressed in the ectoderm of the head. But in no other location. Therefore, as the optic vesicle approaches the overlying ectoderm producing this factor, development of the lens of the eye is induced. Interestingly, induction is not always a one-way path. To that end, differentiation of the lens then triggers the optic vesicle to form the optic cup, which ultimately becomes the retina. This is known as reciprocal development. Most tissues will be exposed to multiple inducers during the course of development. So the optical vesicle stimulates a lens, which then in turn stimulates the optical vesicle to make the optic cup. Should we write that in there? All right. One of the main methods of signaling occurs via the use of gradients. Morphogens, or molecules that cause determination of cells, diffuse through the organism. Locations closer to the origin of the morphogen will be exposed to higher concentrations, while areas further away will have less exposure. Multiple morphogens are secreted simultaneously, resulting in unique combinations of morphogen exposure throughout the organism, which can thereby induce the differentiation of specific cell types. Some common morphogens include transforming growth factor, beta, sonic hedgehog, and epidermal growth factor. include TGF, beta, sonic hedgehog, and epidermal growth factor. Cell migration, cell death, and regeneration. Induction and differentiation lead to the creation of different types of cells. However, these cells are not always in the right location to carry out their function. Further, the sculpting of various anatomic structures requires not only differentiation, but death of some cells. Certain organs also have the ability to recreate injured or surgically removed portions of tissue. Cell migration. Cells must be able to disconnect from adjacent structures and migrate to their anatomically correct location. For example, the anterior pituitary gland originates from a segment of oral ectoderm and must migrate from the top of the mouth to its final location just below the hypothalamus. Neural crest cells also undergo extensive migration. These cells form at the edge of the neural folds during neurolation and then migrate throughout the body to form many different structures, including the sensory ganglia, autonomic ganglia, adrenal medulla, and Schwann cells, as well as specific cell types in other tissues, such as calcitonin-producing cells of the thyroid, melanocytes in the skin, and others. Migration. Cell death. Apoptosis, or programmed cell death, occurs at various times in development. For example, the fingers are originally webbed during development of the hand. The cells of the webbing later undergo apoptosis, resulting in separation of each individual finger and toe. Apoptosis may occur via apoptotic signals or pre-programming. During the process of apoptosis, the cell undergoes changes in morphology and divides into many self-contained protrusions called apoptotic blebs. Apoptotic blebs, which can then be broken apart into apoptotic bodies and digested by other cells, as shown in figure 3.9. This allows recycling of materials. Because the blebs are contained by a membrane, this also prevents the release of potentially harmful substances into the extracellular environment. This is different from necrosis, which is a process of cell death in which a cell dies as a result of injury. In necrosis, internal substances can be leaked, causing irritation of nearby tissues or even an immune response. Necrosis. Apoptosis is when the cell is deliberately broken and digested by other cells. In necrosis, due to injury, if the cell isn't able to do apoptosis, it may just die and have unwanted effects like poisons going to other tissues. All right. Regeneration. Regenerative capacity, or the ability of an organism to regrow certain parts of the body, varies from species to species. Some species, such as salamanders and newts, have an enhanced capacity to regenerate because they retain extensive clusters of stem cells within their bodies. When regeneration is required, these stem cells can then migrate to the necessary part of the body to initiate regrowth. These species are said to undergo complete regeneration in that the lost or damaged tissues are replaced with identical tissues. In contrast, incomplete regeneration implies that the newly formed tissue is not identical in structure or function to the tissue that has been injured or lost. Humans typically exhibit incomplete regeneration in response to injury. However, in humans, regenerative capacity varies by tissue type. Liver tissue has a high regenerative capacity, often able to undergo extensive regeneration following injury or loss. For example, living donors are able to donate up to 50% of their liver tissue because their own livers will regenerate the missing portions. Unfortunately, the heart has little, if any, regenerative capacity, and scarring often results following an injury such as a heart attack. The kidneys have moderate regenerative capacity and are able to repair nephrons after injury to the tubules. However, this regenerative capacity is easily overwhelmed, and kidney failure may result. Or the inducers versus receivers, or I want to call it responder. Senescence and aging. An organism's age changes occur as an organism ages. As organisms age, changes occur in both molecular and cellular structure. This results in disruption of metabolism and eventually death of the organism. Senescence or biological aging occurs in as these changes accumulate and can occur at the cellular and organismal level. At the cellular level, senescence results in the failure of cells to divide, normally after approximately 50 divisions in vitro. Research has demonstrated that this may be due to shortened telomeres, or the ends of chromosomes. Telomeres reduce the loss of genetic information from the ends of chromosomes and help prevent the DNA from unraveling. Their high concentration of guanine and cytosine enables telomeres to knot off the end of the chromosome. Telomeres are difficult to replicate, however, so they shorten during each round of DNA synthesis. Eventually, the telomeres become too short and the cell is no longer able to replicate. Some cells, including germ cells, fetal cells, and tumor cells, express an enzyme known as telomerase. This enzyme is a reverse transcriptase that is able to synthesize the ends of chromosomes, preventing senescence. Telomerase allows cells to divide indefinitely and may play a role in the survival of cancer cells. At the organismal level, senescence represents changes in the body's ability to respond to a changing environment. Aging is complex and often involves not only cellular senescence but also the accumulation of chemical and environmental insults over time. Senescence is aging. Telomeres. What is the difference between determination and differentiation? Determination is is a commitment. Differentiating is when it actually starts changing. What are 3 types of potency? What lineages can a cell of each type differentiate into? Totipotent can differentiate into anything. Pluripotent can differentiate into different things in the layer. So the endoderm can differentiate into either like digestive or respiratory, for example, while the ectoderm will differentiate into either epidermis or neural. Multipotent, on the other hand, will also be able to differentiate into different cell types, but they're further restricted. For example, there are hematopoietic cells that can become blood-related, like white blood cells, red blood cells, or platelets, but they're not going to become anything outside of that branch. What are the 4 types of cell-cell communication? Autocrine, which is to itself. Paracrine, which is when it affects local cells. Juxtacrine is when it only affects adjacent cells. And endocrine, where it can affect distant and targeted cells. What is the difference between apoptosis and necrosis? Apoptosis is programmed cell death, where the cell goes through some process of breaking itself into smaller pieces for other cells to consume. Whereas necrosis is due to injury. The cell doesn't get the chance to prepare its death. And so this can have unwanted effects. Parts of the cell, poisonous parts of the cell, can affect other cells negatively. All right. Fetal circulation. Recall that the placenta shown in figure 310 is the organ where nutrient, gas, and waste exchange occurs. It is crucial that maternal and fetal blood do not mix because they have different blood types. The simplest method to move nutrients and waste products is by diffusion, and this is the preferred method for water, glucose, amino acids, and inorganic salts. Diffusion requires a gradient, so this implies that there is a higher partial pressure of oxygen in maternal blood than fetal blood. In addition to an oxygen gradient, fetal blood cells contain fetal hemoglobin, which exhibits a greater affinity for oxygen than does maternal. This also assists with the transfer and retention of oxygen into the fetal circulatory system. Waste material and carbon dioxide move in the opposite direction. Nice. Blood don't mix. What does mix is water, glucose, amino acids, and salts. Higher pressure in mother, fetal hemoglobin has higher affinity all right. Placental barrier also serves another function, immune protection. The fetus is immunologically naive because it has not yet been exposed to any pathogens. However, accidental exposure can happen in utero. Thus, the crossing of antibodies across the placental membrane serves a protective function. The placenta also qualifies as an endocrine organ because it produces progesterone, estrogen, and human chorionic gonadotropin, all of which are essential for maintaining pregnancy. Let's add antibodies. The umbilical vessels are commonly tested on the MCAT because they demonstrate the need to understand the proper biological definitions of artery and vein. Like all other arteries that carry blood away from the heart, the umbilical arteries carry blood away from the fetus toward the placenta. And like all the other veins that carry blood toward the heart, the umbilical vein carries blood toward the fetus from the placenta. Remember that oxygenation occurs at the placenta rather than in the fetal lungs. Therefore, the umbilical arteries carry deoxygenated blood and the umbilical veins carry oxygenated blood. Let's write down that. Umbilical arteries kind of like pulmonary arteries are also deoxygenated and pulmonary veins are oxygenated because they're going away from the heart and towards the heart, but they're before they are oxygenated, so they're reversed compared to what we're thinking of arteries as being oxygenated and veins as being deoxygenated. There are several key differences between fetal and adult circulation that demonstrate important characteristics of developing organism. The lungs and liver both do not serve significant functions prior to birth. Lungs and liver not functional. Gas exchange does not occur at the lungs, but rather at the placenta. Detoxification and metabolism are primarily controlled by the mother's liver, and nutrient and waste exchange occurs at the placenta as well. Thus, the fetus does not depend on its own lungs and liver. Notably, these two organs are both underdeveloped and sensitive to the high blood pressures they will receive in postnatal life. Thus, the fetal constructs three shunts. The fetus constructs three shunts to actively direct blood away from these organs while they develop as shown in figure 311. I'm not sure I see where the shunts are. But it looks like the umbilical cord's goes right up into the liver, huh? Interesting. Two different shunts are used to reroute blood from the lungs. The first, called the foramen ovale, foramen oval, is a one-way valve that connects the right atrium to the left atrium. This allows blood entering the right atrium from the inferior vena cava to flow into the left atrium instead of the right ventricle and thereby be pumped through the aorta into the systemic circulation directly. Unlike in adult circulation, the right side of the heart is at a higher pressure in the developing fetus than the left side, which pushes blood through the opening. After birth, this pressure differential reverses, shutting the foramen ovale. Second, the ductus arteriosus. Shunts leftover blood from the pulmonary artery to the aorta. Again, the pressure differential between the right and left sides of the heart pushes blood through this opening and into the systemic circulation. Connects the right and the left, the foramen oval connects the right and left atrium. There's higher pressure in the right side of the heart until birth. The ductus arteriosus shunts leftover blood from the pulmonary artery to the aorta. The liver is bypassed using the ductus venosus. Which shunts blood returning from the placenta via the umbilical vein directly to the inferior vena cava. The liver still receives some blood supply from smaller hepatic arteries in the systemic circulation. So this goes from placenta to inferior vena cava. All right. What is the oxygenation status of the blood in the umbilical arteries, in the umbilical vein? Well, the umbilical arteries are deoxygenated and the umbilical veins are oxygenated. What are the 3 fetal shunts? What vessels or heart chambers do they connect? What organ does each shunt bypass? The foramen oval connects the right and left atrium, so it bypasses the lungs. The ductus venosus connects the placenta to the inferior vena cava, bypassing the liver. The ductus arteriosus connects the pulmonary artery to the aorta, also bypassing the lungs, I would assume. Let's double check that. Make sure that's right. Lungs, liver, right. All right, human gestation. Human gestation lasts an estimated 280 days, which are divided into 3 trimesters. As a general rule, the larger the animal, the longer the gestational period, and the fewer the offspring per pregnancy. For example, elephants usually have 1 calf and gestate for 22 months. In contrast, mice have 10 to 12 offspring per litter and gestate for only 20 days. Although you don't need to know every detail of gestation for the MCAT, there are some key developmental events in each trimester with which you should be familiar. First trimester. During the first weeks, the major organs begin to develop. The heart begins to beat at approximately 22 days. And soon afterward, the eyes, gonad, limb, and liver start to form. By 5 weeks, the embryo is 10 millimeters. At 5 weeks. And by week 6, it has grown to 15. The cartilaginous skeleton begins to harden into bone by the 7th week. By the end of 8 weeks, most of the organs have formed. The brain is fairly developed and the embryo becomes known as a fetus. At the end of the 3rd month, the fetus is about 9 centimeters long. At the end of the 3rd month, so that's 12 weeks. Second trimester. During the second trimester, the fetus undergoes a tremendous amount of growth. It begins to move within the amniotic fluid, its face takes on a human appearance, and its toes and fingers elongate. By the end of the 6th month, the fetus measures 30 to 36 centimeters long. Third trimester. The 7th and 8th months are characterized by continued rapid growth and further brain development. Antibodies are transported by highly selective active transport from the mother to the fetus for protection against foreign matter in preparation for life outside the womb. This transfer begins early in the pregnancy, but it is highest in the 9th month, just before birth. The growth rate slows and the fetus becomes less active, as it has less room to move about. Birth, vaginal childbirth, or parturition. Parturition is accomplished by rhythmic contractions of uterine smooth muscle coordinated by prostaglandins and the peptide hormone oxytocin. Prostaglandins, oxytocin. Birth consists of 3 basic phases. First, the cervix thins out and the amniotic sac ruptures, which is commonly called water breaking. Next, strong uterine contractions result in the birth of the fetus. Finally, the placenta and umbilical cord are expelled. These are often referred to as the afterbirth. What are some of the key developmental features of each trimester? First trimester, you got your first heartbeat, 22 days. You got a fetus at 8 weeks. It's about 9 centimeters by 12 weeks. Second trimester, it's 30 to 36 centimeters by 6 months. Third trimester, you get an influx of antibodies to the fetus, and growth slows down. And what are the phases of birth? I didn't actually put the stages, but let's write stages down. First, you got the water breaking. You got the contractions. 3, you got the afterbirth, which is the placenta and umbilical cord. All right. Discrete practice questions. Number 1, which of the following developmental stages has the greatest nuclear to cytoplasmic ratio? Greatest nuclear to cytoplasmic ratio. So the least cytoplasm, the most nuclear, is near the end of like the 16-stage morula. 8-cell embryo, morula, blastula, zygote. Wouldn't it be zygote? Wouldn't it be the later, the more? Oh, zygote is 1 cell. Not zygote. Morula, blastula. Yeah, I'm going to go with blastula because it's later than morula. C. Which of the following associations of a primary germ layer and an adult organ is correct? Endoderm, cardiac muscle. Endoderm, lens of the eye. Ectoderm, fingernails. Mesoderm, lining of digestive tract. I think the only one that's correct is ectoderm, fingernails. 2. C. From which of the following layers does the notochord form? Ectoderm, mesoderm, endoderm, archenteron. The notochord's got to be the mesoderm because the ectoderm makes the neural tube. But the mesoderm makes the notochord. So I'm going to go with mesoderm. 3. B. 4. The influence of a specific group of cells on the differentiation of another group of cells is called competence, senescence, determination, induction. Induction. D. 5. Which of the following is likely to be found in maternal blood during pregnancy? Immunoglobulins produced by the fetus, fetal hemoglobin released from fetal red blood cells, progesterone produced by placental cells, carbon dioxide exhaled from fetal lungs. The lungs aren't going to work yet, so you're not going to have that. Fetal hemoglobin, I don't think is going to cross the border. And immunoglobins from the fetus and the mother is probably not going to happen. Progesterone probably will happen because she's going to have lots of progesterone. Immunoglobulins. What are immunoglobulins? Six, a cell releases a substance that diffuses through the environment, resulting in differentiation of nearby cell. This is an example of what type of cell-cell communication. Through the environment is not juxta, it's para. 6C. 7. A cancer cell is removed from a patient and cultured. The cells in this culture seem to be able to divide indefinitely with no cellular senescence. Which protein is likely activated in these cells that accounts for this characteristic? It's got to be the telomerase, 7D. 8. A tumor is removed from a patient and when investigated by pathologists appears to contain tissue resembling placenta, hair, thyroid tissue, and cardiac muscle. What was the likely potency of the cell from which this tumor originated? Placenta, hair, thyroid, cardiac. So hair is ectoderm. Cardiac is mesoderm. So this isn't even the 3 germ layers, right? This is these have to be totipotent. It's not even pluripotent. These are totipotent. They have to be embryonic stem cells, right? Totipotent, multipotent, pluri, non-potent. Yeah, these got to be totipotent. D. 9. Which of the following is false with regard to adult stem cells? They retain inherent pluripotency if harvested from selected organs. Could be true. They are less controversial than embryonic stem cells. That's also true. They require treatment with various transcription factors. There is a reduced risk of rejection if patients' own stem cells are used. That is also true. It's that C that I just didn't get. They require treatment with various transcription factors. They don't require treatment. What does that mean? Oh, to get them to turn into something? Yeah, I don't know. That's a child is born with an imperforate anus, in which the anal canal fails to form correctly and the rectum is not connected to the outside world. This pathology is most likely accounted for by a failure of cell differentiation, cell determination, apoptosis, neurolation. I think it's going to be neurolation, right? Because neurolation is the stage at which, maybe not. Let's look at their picture. Really, I was thinking about this. But neurolation's close enough, right? Yeah, we're going to go with D, neurolation. Cell differentiation and cell determination, those are too broad. Like, that happens all your life. So it's not specific enough. Apoptosis is cell death. Neurolation is, yeah, a stage of development at least. So 11. Following a myocardial infarction, the heart often heals by the creation of a scar by fibroblasts. This is an example of incomplete regeneration. It's not complete regeneration. It's not competency, and it's not multipotency. If you remember, competency is the ability of a cell to receive signals, inducers, and be, differentiate because of it. So yeah, incomplete regeneration, B. 12. Neurofibromatosis type 1, or von Recklinghausen's disease, is a disorder that causes formation of tumors in multiple nervous system structures as well as the skin. While all cells carry the same mutation on chromosome 17, selective transcription of the genome appears to cause the most significant tumorigenesis in which of the following primary germ layers? It's got to be ecto, because that's both the epidermis and neural system. Nervous system. So, ecto, endo, meso, noto. It's got to be ecto, 12A. 13. Which of the following shows the correct order of early development milestones during embryogenesis? Morula, blastula, grastula. I like that one. C. Let's double check. Oops. Morula, blastula, gastrula. A pregnant woman is accidentally given a single dose of teratogenic drug late in the 3rd trimester. The baby is born 3 days later. Which of the following is most likely outcome? Teratogens are bad things, right? The baby is born 3 days later. Which of the following is most likely to occur? Complete failure of organ development and death of the fetus. Well, development's already happened. Partial failure of organ development with survival of the fetus. Serious disfigurement of the fetus. Respiratory distress at birth but no long-term effects. I don't think you're going to have any failure of organ development. I think you're going to have maybe respiratory distress but no long-term effects. Serious disfigurement, also, I don't know, I don't believe. I'm going to go with D. "15. Which of the following statements regarding fetal circulation is false? In the umbilical cord, there are more arteries than veins." That's true. The foramen oval is the only shunt that connects two chambers of the heart. Now, there's two shunts in the heart. Blood flow in the ductus arteriosus is from the aorta to the pulmonary artery. I would say it's from the pulmonary artery to the aorta, so I'm going to say that's false. The ductus venosus, oh wait, is that, yeah, the ductus venosus is the only shunt that bypasses the liver. That is true. Did I say the right thing for ductus arteriosus? Ductus arteriosus, pulmonary artery to aorta. Yeah. Is the ductus venosus the only shunt? Maybe there's more they just didn't tell us about, right? It says the liver still receives blood supply, but it's the only shunt. So yeah, this is true. So these are all true. Let's double check the right number of veins in the art. I didn't write down that the umbilical cord has a different amount of veins than arteries. Thought I wrote that down. Yeah, I thought they said 2 arteries, 1 vein, right? Oh, maybe I was wrong. Maybe it's 1 artery and 2 veins. Umbilical cord. 2 arteries and 1 vein. So this is true. The foramen ovale is the only shunt that connects 2 chambers of the heart. That's false. There are 2. So that, all right, 15 is B is the one that's false. All right. This is true that the blood flow is from the blood flow in the ductus arteriosus is from the aorta to the pulmonary artery. No, that's also wrong. It's from the pulmonary artery to the aorta. It's from the aorta to the pulmonary artery. It says pulmonary artery to aorta. That's what I wrote down. I think that's what they said. Leftover blood from the pulmonary artery to the aorta. So this is true. Oh no, this is false. 15C is definitely false. So I'll change that to C. All right, let's see how we did. 1C, 2C, B, 3B, 4D, 5C, 6C, 7D, 8D, 9 is A, not C. Embryonic stem cells are controversial because they require destruction of an embryo to harvest, eliminating choice B. Adult stem cells are significantly less controversial but require treatment of various transcription factors in order to induce an increased level of potency, eliminating choice C. Rejection. Oh, they do require transcription factors in order to induce an increased level of potency. Eliminating choice C. Rejection is a concern when foreign cells are introduced into an individual. Using one's own stem cell should remove this risk. Eliminating choice D. Adult stem cells are not naturally pluripotent unless pluripotency has been induced by strategic use of transcription factors. Therefore, choice A is the correct answer. So they retain inherent pluripotency only if they're induced pluripotency. They don't retain inherent pluripotency. So that is false. And C, I thought, which was false. This is true. They do require treatment with transcription factors. It's how they induce pluripotency. 10 is C, not D. During development programs, cell death occurs in multiple locations in order to ensure development of the correct adult structures. One of the places in which this occurs is between fingers and toes, others in the digestive tract where a central lumen is formed. If apoptosis did not occur correctly in the digestive tract, an imperforate anus would result. Failure of determination of or differentiation would likely result in the absence of and anorectal structures altogether, eliminating choice A and B. Failure of neurolation would lead to an absence of a nervous system and would not be compatible with life, eliminating choice D. So, C, removal of the webbing is similar to, removal of the hole for the anus. And if that doesn't happen, you don't have an anus. All right, 11B, 12A, 13C, 14D, 15C. All right. Well, that is it for embryogenesis and development. Next time, Chapter 4, the nervous system. Thanks for watching. See you next time.